मिनिमाइज करने का बात
पूर्ण झाल्यानंतर चेक करू. Good afternoon, students. Today we'll take up lesson number 9. It's control and coordination. Students, I have divided this lesson into 3 parts. So we'll be taking the first part today. Okay. Now let's have a brief introduction about this. What is control and coordination? Linking together in time and space of various activities in an animal is called coordination. So what is coordination? It is linking together in time and space of various activities in an animal. And this is brought about by nervous and endocrine system. Together these two systems, they are referred as neuroendocrine system. Nervous system is based on the special properties of sensitivity, conductivity and responsiveness. It is ectodermal. Nervous system is ectodermal in origin. Nervous system is fast acting. Effects are localized and involves electrical and chemical transmissions. The endocrine system is slow acting. Effects are diffuse type and relies on chemical transmission through blood. So understand the difference between two, that is the nervous system and the endocrine system. Nervous system is fast, endocrine is slow acting, and the effects in nervous system are localized and involves electrical and chemical transmission. Right? Whereas in case of endocrine system, the effects are diffuse and they rely on the chemical transmission through blood. Then plants and animals both show control and coordination mechanism. In case of plants, by sending chemical signals which brings various movements like phototrophic movement, chemotrophic, geotrophic, that is towards the earth surface, towards the soil, then hydrotrophic and hypnotactic movements. So these movements are seen in case of plants. In case of animals, showed there is a gradual increase in the complexity of their control and coordination by giving electrical and chemical signals. Then, now let's understand about the sponges first. Sponges, they are the only multicellular animals without a nervous system. Imagine students, sponges are the only multicellular animals without a nervous system. They belong to the phylum Porifera. Porifera means they bear lot number of pores. You can see here in the diagram given, see so many pores are present over here. Okay, these are known as the ostia, and this main one, it is known as the osculum. They have specialized cells that carry out different functions within their body. Sponges have no nerves or brain. Beginning itself, I told that they do not have a nervous system. They do not have nerves, they do not have brains, nothing. So they lack any ability of cognitively control their movement. However, touch or pressure to the outside of a sponge will cause a local contraction of its body. Like if if anybody touches this, what happens? It will contract the body. This was sensitive one. Adult sponges are not mobile, but they are able to react to certain physical stimuli despite lacking conventional organs and sensory system. Many sponges protect themselves by producing toxins that make them poisonous to the immediate or to the primary predators. Next you see nervous system in Hydra. Hydra, it is a cnidarian. See, its spelling is starts with C, but C is silent over here, so we pronounce it as cnidarian. Hydra is a cnidarian, is a simple multicellular organism. It possesses one of the anatomical simplest or most primitive nervous system, which is a diffuse nerve net spread throughout the body with no signs of centralization. Students, when you see this diagram here, you see this is Hydra. You can see that there is no centralization. That means you see, like in human beings, for example, we have brain, right? There is nothing over here in this way, right? No brain, no ganglia, nothing is present over here. Only it is a network of the nerve fibers throughout. And what happens? The sensitive but sensory cells, they are present at the tentacles and extra and the epidermis and gastroid epidermis. So if slightly if it is stimulated, the whole body will show the reaction of it. Okay, this is about the nervous system in Hydra. That is, it does not have anything as a centralization or a central nervous system. Yeah. So we can further say that which is a diffuse, so we call it as a diffuse nerve net, which spreads throughout the body with no signs of centralization. No no sign of centralization means a loosely organized system of nerves with no central control at all. Therefore, diffuse type it is, right? It is made up of about 6,000 nerve cells belonging to two morphological types, that is sensory and ganglion neurons. And what is ganglion neuron, student? Ganglion means what? It is a group of neurons, cell bodies. That is a cycle. The neurons constantly develop from the multipotent interstitial stem cells. So these neurons, they develop uh, continuously, that is constantly, from the multipotent interstitial stem cells, right? The nerve cells are scattered throughout the body and interconnected to each other by synapses between the fibers to form the nerve net or net plexus between epidermis and gastrodermis. Just now I told you, I showed you the diagram that these nerve cells, they are the sensory cells, they are present at the epidermis, the tentacle part, at the tentacles and the remaining part of the hydra and in gastrodermis. Most synapses are electrical. Neurites, what are neurites? These are the neural processes, okay, are located in the ectoderm and the endoderm. It detects the stimulus from internal and external environment. In other words, simply to say, student, that messages are received or detected from point of stimuli, and the message is diffused in all the direction in hydra. That is, stimulation at any point spreads to cause movement of the entire body. The nerve impulse shows no polarity or direction. That means what, students? Suppose you see this diagram now. Suppose there is a stimulation given over here. The whole body will show its reaction. Okay, when stimulus is given, the impulse will be circulated in the whole body. There is no particular direction that it will move upwards, up first, or second, or downward, or any other side. Nothing like that. It will spread throughout the body. So this is the nerve net. Okay, there is no centralization. Now you can see over here, the nerve cells are present. These dark structures you can see, student, this is the nerve cells, okay? And you can see these thread-like structures in between. These are the fibers, the processes that originate from the cell, okay? And they are referred as the neurites, right? Then you see these nerve cells, they form the network. These are the tentacles, okay? I think this much is only required for to study the nervous system in hydra. Next you see the nervous system in planaria. Planaria, it is a flat form and it belongs to the phylum Platyhelminthes. You can see the diagram over here. Okay. Nervous system of planaria is not very complex. It's very simple, students. It is having a nerve cord, ventral nerve cord. See, this is the ventral nerve cord. You can see in the diagram this elongated structure. It is ventral in position, so ventral nerve cord. These two, they merge or they merge together at the anterior part, and this is then known as the brain. This forms the brain part. Okay, that is, they converge and they form a rudimentary brain waveform. This is the eye spot, clear, and these are the transfer nerves or the commissures. These are the lateral nerves over here. So internally, when we see, student, we find that it looks like a ladder. You can see the diagram over here. Internally, you can see that it looks like a ladder. Right now, let's understand this more clearly. Statement form. Nervous system of planaria is not very complex. The planarian CNS, that is central nervous system, is located on the ventral side of the body. It is composed of a mass of cerebral or cephalic ganglions in the head region. See here, this part, right? And a pair of ventral nerve cords. It is abbreviated as VNC, that is ventral nerve cord. Right? These nerve cords, they converge to form a rudimentary brain. See this part over here, right? Eye spots are also seen. Further, the nervous system of planaria, it looks like a ladder within the body, longitudinal. See, this is the longitudinal nerves and these are the transverse nerves. So it looks like a ladder or a staircase. VNC, that is ventral nerve cords. It picks up impulses from muscles and sends message to the cerebral ganglion, right? And for what cerebral ganglion does is it is a simple brain which can detect the light, it can detect movement, it can detect the touch, it can detect the water current and also the chemicals. This is about the nervous system in planaria. Next comes the neural tissue. Now neural tissue includes the neurons or the nerve cells and neuroglia or the glial cells. See, neural tissue we are talking about, students, okay? So it consists of neurons, right? And neuroglia cells. Yeah. So neurons are the structure and functional unit of the nervous system. You can see here, you have already learned this in the lower classes. See here. See, this is a neuron. This is a neuron. We'll come back again later to understand this in detail. So neurons are the structure and function unit of nervous system, and whereas the neuroglia cells provide nourishment to the nerve cells. Understand this. Neurons are also otherwise known as the nerve cells. Okay, and neuroglia cells are also known as the glial cells. Right. So this neuroglia cells, what is the function of the nerve cell or the neuron? It is a structure and functional unit of the nervous system, and these neuroglia cells, they provide nourishment to the nerve cells or to the neurons. Okay? Protoplasmic extensions of the nerve cells form the neural pathway called nerves. Nerves works like electricity wire. Average speed of nerve impulse is 130 meter per second. Nervous control takes place through electrical signals called nerve impulses. Impulses are either sensory or they can be motor. If they are sensory, they are referred as afferent, and if they are motor, they are referred as efferent. See the spelling A and E. Okay. Sensory impulses are transmitted, that is afferent, by sensory, that is afferent nerves from the sense organ to the CNS. So sensory impulse is transmitted from the sense organ to the central nervous system, that is CNS, and vice versa in case of motor impulses. Okay, they are transmitted from the nerves, from the motor nerves, that is efferent, from central nervous system to the effector organs like muscles and glands. So I think you have understood this. What is this neural tissue consisting of neurons and the neuroglia? Okay. Next, the neuron or the nerve cell. So there we studied about the neural tissue, which consists of the neuron or the nerve cells, right? So, and neuroglial cells. First we study about the nerve cell or the neuron. Now, a neuron has three parts basically. One is cell body, or it is otherwise also known as cyton. And this cyton or the cell body contains a centrally placed nucleus, sorry, neuroplasm, then nasal granules, sorry, nasal granules and two extensions. I just repeat, the cell body or the cyton, it contains nucleus, okay, and neuroplasm. It consists of nasal granules and two extensions, that is in the form of dendrites and the axon. Then the second part is the dendrons. These are short and numerous. They receive information from other neurons and sensory receptors. Then third part is the axon. It is single and long fiber. Remember the difference between dendrites and axon, student. Dendrites, they are short and they are numerous, and they are the receiving cell. Okay, they are the receiving branches or the processes. Here, whereas axon is a single and it is a long fiber, and it transmits, it works as a transmitter of the message from the cell body to the neuron or the muscles or glands to the next one, to the next neuron. Okay, so it is a single and long fiber. It passes or transmits messages away from the cell body to other neurons, muscles or glands. Next is myelin sheath, students. Myelin sheath, it is produced by, or it is formed by a cell which is present around, which is present on either side of the axon. So these cells are known as Schwann cells, and they have a nucleus known as the Schwann nucleus. Okay? The function of this cell is to produce a fatty substance known as myelin, right? Myelin, and this covers the axon, but it does not cover the axon completely or continuously, right? In between, it is interrupted, right? So this interrupted part is known as node of Ranvier. Simply to say, node of Ranvier does not have a myelin sheath covering. Okay, now let's understand again. It is formed of by Schwann cells. Myelin sheath is a protective covering for axons. It is an insulating layer. It helps speed neural impulses. It also it allows electrical impulses to transmit quickly and efficiently along the nerve cells. Myelin does not cover the axon continuously. As I told, it is interrupted in between. The bare part of axon, that means one which is devoid of myelin, is known as the node of Ranvier. Yeah. Terminally, the axon gives out branches and they are known as the telodendrite. And these terminal buttons form in, form in the junctions with the other next neuron. Here you see. Now we'll talk about the nasal granules which are present in this over here. You see here. Now let's understand this diagram first. See, this is a structure of a neuron. Okay, this is the structure of a neuron. Here you see, or the nerve cell. It consists of a cell body and a elongated process. This cell body gives out two different types of process. One which are short, they are known as the dendrites, right? And the one which is single and it is elongated, this is known as the axon. Axon is covered by a myelin sheath membrane produced by the Schwann cell. Okay, see here. But this membrane, this myelin sheath is not continuous. See here, it is interrupted in between, and this is known as the node of Ranvier. This axon ends up into the terminal part, that is the axon terminal, which is having a synaptic knobs like structures over here or terminal buttons. Yeah. Now inside the cyton, there is, see this cytoplasm over here, this is known as the neuroplasm, embedded within which are present minute granules known as the Nissls granule and a nucleus over here. I hope you have understood the structure. Now see, what are Nissl granules? These are of rough endoplasmic reticulum. These granules are very useful for protein synthesis. We have also studied in protein synthesis process about endoplasmic reticulum. Rough endoplasmic reticulum, RER. So Nissl granules are the part of it. Okay. Then depending upon the presence and absence of myelin sheath, this myelin sheath I told you that not all nerve fibers or not all axons, they are enveloped or they are covered by the myelin sheath. Those which are covered, they are known as the medullated nerve fiber, and those which are devoid of the myelin sheath and is covered only by the neurolemma, they are referred as the non-medullated nerve fiber. So depending upon the presence of myelin depending upon the presence and absence of myelin sheath, the nerve fiber or the axon can be classified as the medullated and non-medullated nerve fibers. Now here, again, depending upon, I told you in this diagram over here, this cyton or the cell body gives out number of processes. Two different types of processes. One which are mini, okay, and they're short. And the other one which is single and elongated. Okay, now depending upon the nerve cell or the neuron, that how many processes are present, it is classified as unipolar, bipolar and multipolar. Okay, multipolar is missing over here and multipolar. So depending upon the number of processes, neurons are classified as unipolar. That means only one process is there. See, from cell body only one process is starting. Okay, is originating, and bipolar means two are there. See, this here is one and here is one, and multipolar, the one which studied the structure, multipolar. Okay, this is the multipolar where the number of dendrons are seen and the elongated axon is seen. Now here, students, I told you about the neural tissue, which includes the nerve cells or the neurons and the neuroglial cells or the glial cells. Okay, so we have completed with this neuron, understanding the information about the neuron or the nerve cell. Now we switch on to the next type of neural tissue, that is the next type of cell of neural tissue, that is neuroglial cells. Right? These are also known as the glial cells. They are glial, means the glue cells. Okay, They are the helper cells or the supporter cells. These are non-neuronal cells. That means they are not having in the CNS. They are not having the neuron. Okay, so they are non-neuronal cells. We say it. That is in the CNS. CNS means brain and spinal cord. Okay, and the peripheral nervous system. So these cells, that is neuroglia cells, they are present in the CNS as well as in the peripheral nervous system. Right? The number of glial cells are more than the neurons. Okay, student, the number of glial cells are more than the neurons. They maintain what is the function of these glial cells now? They maintain homeostasis, right? They form myelin and provide support and protection for neurons. So I told in the beginning that neuroglial cells, they are the helper cells. Help whom? It helps the neurons or the nerve cells. They also are involved in the nutrition and maintenance of the nerve cells. Neuroglia in the CNS include the following cells here. You see, the neuroglia in the CNS, that is central nervous system, includes these cells, that is astrocytes, microglia, then ependymal cells, and then oligodendrocyte cells. These four different types of cells. They are present in the CNS. These are all types of neuroglia cells which are present in the CNS, that is central nervous system. I also said that they are also present in the peripheral nervous system. So then these two types of cells are there. One is satellite cell, another one is the Schwann cell. Let's understand the function of each such type. Now here you see. First, let's see here, the neuroglia cells are there. This is a chart, student, showing the function of each cell over here. Neuroglia cells are there. Where they are found? They are found in the peripheral nervous system and they are found in the central nervous system. Okay. First, let's understand what are the different types of neuroglia cells which are found in the peripheral nervous system. One is the satellite cells. Okay. What is its function? Its function is it surrounds neuron cell bodies in ganglia. Okay, it regulates oxygen, carbon dioxide, nutrient and neurotransmitter level around neurons in the ganglion. So it is present around the cell bodies in the ganglion. Which cells? Satellite cells, right? Then another peripheral neuroglia cells is the Schwann cells. Where it is present, it surrounds axons in PNS, that is peripheral nervous system. They are responsible for myelination of peripheral axon. Myelination means to form the myelin sheath over here, over the axon. Then it participates in repair process after injury. So if injury takes place, the Schwann cells also helps in the repair process. Right? Now this was about the peripheral nervous system. Neuroglia cells present in the peripheral nervous system. Now let's understand the neuroglia cells present in the CNS, that is central nervous system. First of all, let's take up this oligodendrocytes. Very easy to pronounce and very easy to write the spelling also, student. Oligodendrocytes, right? Sites indicates for the cells. Then myelinate CNS axon. So what does it do? It helps in forming the myelin sheath, right? And it provides structural framework also. And which cells these are? These are oligodendrocytes. Clear? Next is astrocyte. They maintain blood-brain barrier, then provide structural support. It provides structural support, then regulate ion, nutrient, and dissolved gas concentrations. It absorbs and recycles neurotransmitters, then finally forms scar tissue after injury. That is, if injury takes place, a scar formation is done with the help of astrocytes. Next you see the third type, that is the microglia. It removes cell debris, waste and pathogens by phagocytosis movement or nature. The microglia cell, they are phagocytosis. Okay. Then last neuroglia which is present in CNS is ependymal cells. Right? It lines ventricles of the brain and central canal, that is the spinal cord. Okay, student, brain, we will be studying further the ventricles of brain. Brains are having cavities inside which is known as the ventricles. Okay, so these ependymal cells, they line up the ventricles of the brain and the central canal of the spinal cord. Moreover, they also assist in producing, circulating and monitoring of cerebrospinal fluid. So cerebrospinal fluid is present, which is again assisted by the ependymal cells. So this is about the neuroglia cells. and the functions. Now next, synapse. What is synapse? Student, between two neurons or two nerve cells, there is a gap in between, okay? And that is where the exchange of the message or the impulse take place from the axon of one uh, neuron to the dendrite of the next neuron. Okay, so let's understand. It's a junction between the axon terminal of one neuron and the dendrite of the next neuron. It is separated by a small gap called synaptic cleft. The transmission of nerve impulses is carried out by chemical like neurotransmitters. Right? This is about the synapse. Okay. Now let's first understand the properties of the nerve fibers. So these are the various properties, that is excitability or irritability, then conductivity, stimulus is there, then summation effect is there, all are non-law, then refractory period is there, synaptic delay is there, synaptic fatigue. Let's understand one by one in detail now. Now what is excitability? It is the irritability, it is also known as the irritability, okay? It is the property of a neuron to produce an action potential. This is a function of permeability and the chemical and electrical state of the neuron cell membrane. The intensity of electrical stimuli influences the excitability of the neuron. Then the conductivity. Now this impulse is there, excitation is taking place, that is impulse is there, and now it has to be conducted. Okay. So the next character property is the conductivity. It is an ability to transmit the impulses all along the whole length of axon without any change in the amplitude of the action potential. Okay. Then next is the stimulus. Next is the stimulus. It is it is any detectable physical or chemical electrical change in the external or internal environment which brings about excitation in a nerve, muscle or organ. Stimulus means to disturb something, okay? Physical disturbance to any structure, any organism. That is external or internal. It can be internal also. The minimal effective, that is the threshold, stimulus is adequate only for fibers of high excitability. Right? So if that particular only for fibers of high excitability, that means the minimum, that is the threshold stimulus is also enough for getting stimulus is enough for to the cell or the fiber to get excited or irritated. But a stronger stimulus excites all the nerve fibers. The receptors transform environmental stimuli into electrical nerve impulses. These impulses are then transmitted via neurons to the CNS where decisions making occurs. Okay, so student, what we have seen, excitability, the stimulus. This thing, it is getting an impulse, then this impulse has to be conducted, that is stimulus. If it is of minimum effect also, it is adequate enough or it is enough for the fibers which have high excitability quality. Okay, but a stronger stimulus excites all the nerve fibers. Now the next property, these are the three different, that is excitability, conductivity or stimulus. Next one is summation effect, that is the addition. Okay, summation means the addition. It is about adding up the effect of multiple stimuli that are all individually sub-threshold so that together they are supra-threshold and are responsible to generate an action potential. That means come in response, that is summation effect. Then all are non-human. It is the principle that the strength by which a nerve or muscle fiber respond to a stimulus is independent of the strength of the stimulus. If the stimulus exceeds the threshold potential, the nerve or muscle fiber will give a complete response, or otherwise there will be no response. That means what all or none will be, suppose if the stimulation is given, the strength is enough, that is the threshold potential, then it will complete the response, or otherwise there will be no response at all. Clear? The next one is refractory period. In this, the neuron cannot be excited to generate a second action potential, no matter how intense the stimulus is. Okay, whatever may be the intensity of the stimulus, it will not get excited to generate a second action potential. This is refractory period. Then synaptic delay. Yes, it is the time interval between peak of inward current through the presynaptic membrane and commencement of inward current through the postsynaptic membrane. The time taken by impulse to cross a synapse is about 0.5 milliseconds. That is the synaptic delay. The last one, that is the synaptic fatigue. Fatigue means tired, exhausted. Okay. So it is a short-term synaptic depression. The transmission of nerve impulse across the synapse halts temporarily due to exhaustion of its neurotransmitter. That means the neurotransmitter got exhausted. So for a fraction of millisecond, it will stop. Okay. That is the synaptic fatigue. Next is types of synapse. Now, types of synapse, they are the electrical synapse and the chemical synapse. The functional gap, understand this now properly, student. The functional gap, that is the region of communication between two neurons, is called synapse, that I told you in the beginning. Okay, the nerve cell contributing the transmissional terminal is known as the presynaptic neuron, and the one which contributes to the generator region is known as the postsynaptic. Yeah? Then intercellular space between two neurons is called synaptic cleft. I have already told you all these things, just repeating to introduce you the two different types of synapses, that is the electrical synapse and the chemical synapse. Right? Then intercellular space between the neurons is called the synaptic cleft. I told you this may be around 20 to 30 nanometers distance. Right? There are two types of synapses. One is electrical synapse and the other one is the chemical synapse. Now electrical synapse, in this what happens, in this the pre and post synaptic neurons are in very close contact. The electrical current flows directly from the neuron to the other, occurs across the synapse. Okay, the electric current flows directly from one neuron to the other across the synapse. Clear? The transfer of impulse is faster when compared to chemical synapse. So now see, electrical synapse, I told you. So it is the transfer is faster when compared to the chemical synapse. Yeah. Now let's understand what is chemical synapse. In this, the presynaptic membrane includes the following structure, that is the synaptic button or the bulb, then a synaptic cleft, and the postsynaptic membrane. The neurotransmitters transmits the impulses at these synapses. The synaptic bulb, that is the button, possesses synaptic vesicle filled with acetylcholine. This is the neurotransmitter, student. This is the chemical acetylcholine which is present in these bulbs or in these buttons. Okay, and they are known as the neurotransmitter. When the impulse arrives at synaptic bulbs, this neurotransmitter is released in the cleft, which in turn stimulates the postsynaptic membrane. The neurotransmitter is immediately removed from the synaptic cleft by an enzyme. So once the message is transferred, students, or transmitted, then what happens? It is immediately removed from the synaptic cleft by an enzyme known as choline sterase. Okay, there is an enzyme known as choline sterase which brings back the neurotransmitter back to the bulb once the message is transmitted, and it makes the synaptic cleft ready for the next impulse. Yeah. Next is the transmission of the nerve impulse. Now transmission of the nerve impulse. Here the extracellular fluid which surrounds the neurons have property of excitation. Their membrane is selectively permeable to different ions, that is cations, potassium ions and sodium ions. When the neuron is not conducting any impulse, that is when it is in the resting state, it is in a polarized state. And at this time, the interior of axon has less cations. Therefore, more electronegative as compared to ECF. What is ECF, students? It is extracellular fluid compared to ECF with more cations, hence electropositive. Okay. The next one is the on stimulation. Now what happens on stimulation? That was the resting phase. Now what happens on the stimulation? the membrane becomes freely permeable and causes the rapid influx of the sodium ion. This makes the inner side positively charged now, and this, when it becomes positively charged, it is known as depolarization. Right? Depolarization is the action potential. During this phase, when axon is excited by a stimulus, neurilemma undergoes depolarization. The polarity of the membrane gets reversed after excitation because sodium ions move inward and potassium ions they move outward. The wave of depolarization and action potential is carried along the whole length of axon membrane. Now next is the third part is the recovery phase, or it is also known as the repolarization. Okay, the charge of polarity results in potential differences at the site and the axonal end. Hence, the local circuit is formed and current flows from dendron to axon. Similarly, sequential local circuits are formed along the length of the axon and the impulse is conducted along the length of a nerve fiber. The stimulus inducing permeability to sodium is short-lived. Sodium ions is short-lived. Once the impulse is conducted, the membrane is repolarized. The fiber becomes responsive once again for further stimulation. So I hope you understand the meaning of polarization. It is the resting phase. Okay, during this phase, outer surface of neurolemma is positively charged and inner surface is negatively charged. Sodium ions are present in high concentration outside the membrane and in low concentration on the inner side. So this is during the resting phase. Then during the depolarization phase or the action potential, what happens? Axon is excited by a stimulus, neurilemma undergoes depolarization. The polarity of membrane gets reversed after excitation because sodium ions move inward and potassium ions move outward. The wave of depolarization and action potential is carried along the whole length of axon membrane. And then comes the repolarization, that is the recovery phase. Now it has to again, it will recover, it means it will recover again for the next impulse. Okay, during this phase, membrane allows diffusion of sodium ions move fast to outside and potassium ions start moving inside. So again, positively charged, negatively charged, it happens like that, and sodium moves outside, then potassium will move inside and vice versa. So move faster outside and potassium ions start moving inside. Therefore, outer surface of neurolemma again becomes positively charged, and inner surface becomes negatively charged. The nerve fibers becomes ready for the transmission of the next impulse. Okay, student. So this was what I wanted to tell you today, right? I stop here. Any clarification you want, anything which is not clear to you, you can ask me, contact me on WhatsApp. You can ask on WhatsApp also, or you can make a phone call also. Okay. So I stop here. Thank you so much.